Namaste. Welcome to Dr. Varsha Gaitonde's channel. In this lecture, I am going to talk about tilling, ecoTILLING and deTiLLING. We'll go one by one. First, we'll talk about tilling. Tilling is a general reverse genetic approach. As you know, forward genetics means first we'll start with phenotyping. We'll go to the field. We'll take the observations like plant height, color, any kind of phenotypic means observable observations. We'll record them. Then what we'll do? We'll go for PCR. We'll just see the DNA sequence. Then we go for genotyping or DNA sequencing, whatever it may be. But reverse genetics means it is just opposite to that. That means first we'll take the DNA sequence, then we'll go for protein analysis, and from that we will go for phenotyping. Means the results what we obtain from the protein sequencing or DNA sequencing, we then compare with the phenotyping. That is the procedure of reverse genetics. So I was talking about tilling. Tilling is targeted induced local lesions in a genome. In this technique, this combines the chemical mutagenesis with PCR based screening to identify point mutations in regions of interest. That means tilling is an approach. Three things you need to remember here. It is a reverse genetic approach. This is an approach for chemical mutagenesis. That means for creating the mutations or finding out the mutations in the population. Then this combines chemical mutagenesis and PCR approach. And this is mainly for identifying the point mutations. Tilling is a powerful technology that employed heteroduplex analysis to detect which organism in a population carry single nucleotide mutations in specific gene. Heteroduplex analysis means this is an approach to identify the SNPs or single nucleotide changes. Tilling can also be used to detect naturally occurring SNPs in gene among the accessions variety or cultivar to study the gene functions or detect the genetic marker in population. We'll see some of the historical prospects of tilling. Tilling was first discovered in or first approached in 1990s by McCollum, who worked on characterizing the functions of two chromomethylase genes in Arabidopsis. He worked how he utilized this technology means like tDNA lines and antisense RNAs he has taken but was unable to successfully apply these approaches to characterize CMT2, that is a gene. The approach that was successfully turned out to be what is known as tilling. This combines the standard and efficient techniques of mutagenesis as I talked already. The technique is used for mutations, identifying mutations. The main steps involved here are development of mutagenized population, then DNA preparation and pooling, and finally mutations discovery. As you know, I just tell you the approaches which have been followed for identifying the functional genes. The approaches are RNAi, gene knockout, site-directed mutagenesis, and transposon tagging. Coming to the methodology involved in tilling. First we'll discuss in figure in the later slides, but now I will tell you the bullet points of methodology of DNA tilling. Here development of mutagenized population, then EMS mutagenesis, that is ethyl methane sulfonate, then development of M2 population, DNA preparation and pooling of individuals, mutation discovery, PCR amplification of a region of interest, then mismatched cleavage, detection of heteroduplexes as extra peak of band through HPLC or acrylamide gel, then identification of mutant individual or phenotypic recovery, then sequencing of the mutant product. These steps you need to remember in any of the exams. They may ask the procedure. That means for 5 marks, these bullet points along with some explanation is enough. This is the procedure as I have already told you the 3 steps. One is mutagenesis and development of mutant population. Second is DNA extraction and sample pooling and detection of mutations. Here in this picture you can clearly see first the M0 generation is here. From there the EMS induction is given. M1 and M2 generation. The phenotyping can be carried out either in M2 or M3, because in M2 if enough variation is there, then it is quite difficult. After attaining some kind of homogeneity or homozygosity only we can go for the phenotyping approach, so that the identification of target traits will be easy, or the gene of interest identification will be quite easy. After M3 phenotyping, M4 seeds are taken, means from many plants the DNA is pooled, okay, and that is just placed in the wells. That is the second step, sample pooling. In the third step here, that is detection of mutations. Here the strands or DNA is taken. Heating or cooling means that is done under PCR condition, polymerase chain reaction. Here homoduplexes and heteroduplexes are formed, okay. Whenever the mutagenesis is exposed or mutations are induced in a population, we may find homoduplexes and heteroduplexes. Heteroduplexes are where the mutations are induced or mutations are found, okay, or variation is found. Then cell 1 digest. Cell 1 is means celery 1, the enzyme taken from celery. This enzyme which gives a slant cut where the heteroduplexes are formed, so that whenever we go for the gel electrophoresis, after this homoduplex heteroduplex formation step or cutting the heteroduplex through cell 1 digest, means that is not only given to heteroduplex, for homoduplex also digestion will be given, but due to the straight strands there won't be any cut. But wherever heteroduplexes are found, cut is induced. Then directly we'll go for gel electrophoresis, where the bands are placed, means homoduplex and heteroduplex. Until that we don't know, because we have not seen what kind of bands we obtain. So the digested PCR product is ran in this particular gel slab, or we can say the gel electrophoresis. After running that, we will just take that particular band and we'll just see sequence, means at this stage we can identify the homoduplexes and heteroduplexes based on the intensity of bands as well as the illumination. And after that, whenever we go for sequencing, we can easily identify the heteroduplexes or the mutations which were detected in the population. This is all about the approach of tilling. This is how the tilling protocol goes. After inducing the mutagenesis, when the homodimer and heterodimer or homoduplex or heteroduplexes are formed, the forward and reverse primer designing is also important. That should be designed very carefully, and the sequence of that will entirely be changed. Let us move to some of the chemicals used in this particular technique. The first chemical is ethyl methanesulfonate. As I told you, this is used for inducing the mutagenesis. This is a common alkylating agent. Here the mode of action is by transferring their alkyl group at 6 oxygen and 7 nitrogen to the DNA base and the phosphate group. This produces transition mutations. As you know, there will be transitions and transversions, but here transitions are formed from GC to AT because it alkylates the G residues. Also, the ethyl nitrosourea used for induction of mutations, it functions just like ethyl methane sulfonate. Then the second chemical is cell 1 endonuclease. As I told you, this is an enzyme taken from celery. S1 nuclease, this is generally called as The family of single-strand specific nuclease specially recognizes mismatches in the heteroduplex. This cleaves DNA on the 3-end side of the mismatch single nucleotide polymorphisms. This I have explained you already. For better understanding, just refer this figure. Next is choosing the target sequence. The optimal length of target sequence that can be TILLed in a single reaction is 1500 bp. But most eukaryotic genes are over 2000 bp and sometimes much longer. Since the objective of TILLing is to identify plants with deleterious mutations in the target gene, and most mutations in non-coding sequence such as introns, untranslated regions and promoters will have no effect on gene function. So the target sequence should be chosen to minimize such sequence and maximize the coding regions. Means our TILLing approach should be focusing or targeting to the coding regions, because we may find the expression sequences known as exons at that particular coding regions. Some of the applications of TILLing we shall discuss here onwards. TILLing could become absolute when a general technology for easy modification of target genes will become available. This verifies the role of particular candidate genes identified during QTL mapping. TILLing approach was practiced, or it was experimented in tobacco to reduce the narcotin content, means nicotine content, which is derived from nicotine by N-demethylation. Its presence is undesirable in cultivated tobacco because it's a precursor of carcinogen N-nitroso nicotine. This narcotin is minor alkaloid is most commercial tobacco varieties. However, phenomena known as conversion result in the progeny of low nicotine to narcotin converting parent plants having much higher conversion rate of nicotine to narcotin. That means narcotin is a poisonous or it is a hazardous carcinogenic compound which is produced by nicotine. By using TILLing approach or by using targeted mutagenesis approach, the conversion of nicotine to narcotin was prevented or prohibited. It is particularly useful for quality traits and recessive genes that are difficult for breeders to select in the field. In the coming years, numerous examples of TILLing can be found in the breeding approaches. TILLing provides means to address limited nucleotide diversity of wheat and other crops, especially when favorable deletion alleles are not available, or when genetic alterations other than knockout alleles are derived. That means you may be knowing that NGS technology is very helpful in identification of the rare alleles. Through SNP technology, that is single nucleotide polymorphism, we may find the rare mutations or rare changes between two individuals, but it requires sequencing. In TILLing step, after inducing the mutagenesis, finally we may go for the sequencing step, so that identification of particular gene of interest or inducing the mutations, or we may say the genetic modifications which we induce artificially, that is a different approach, because in genetic modification we should undergo many of the regulations, but if we follow the TILLing approach, there are very less regulations, and the induction of changes is also quite easy. TILLing technology used to identify genetic diversity in homologous genes is exceptionally well suited to polyploids. Targeted genes of interest through TILLing allows development of novel traits and functional markers simultaneously, because of mutations serve as a landmark that can be converted into molecular markers. That means making or generating molecular markers through TILLing approach is also a possibility, because at the end of the tilling technology we go for sequencing. By using that particular sequence we may go for designing the forward and reverse markers for a particular gene, or it is a best approach when the SNPs are identified. This can also be used to develop the functional markers. The use of tilling also allows surveys of naturally occurring variation to examine thousands of individuals in a mapping population or thousands of different varieties at a time. This approach is especially useful when no naturally occurring or very limited nucleotide diversity is available. Tilling allows identification of new alleles that don't necessarily have knockout gene function and that can be identified in the same genetic background. Because an allelic series of point mutations can be identified with different potential effects on protein function, ranging from null to reduced function to dominant effects, a range of phenotypes may be possible. Tilling was used in many of the plants. It is being used in different kind of genera, even the species Arabidopsis, rice, lotus, and even for the particular traits like nitrogen fixation and the functional role of sucrose synthase was the target of lotus, and it was also used in different other crop plants. Coming to utility of tilling, this is a high throughput, sensitive, cheap, rapid reverse genetic tool. By using this, target gene can be investigated for each domain or even each single amino acid. Particular allele identified by a breeder on a trait associated gene is confirmed to useful or not by using tilling or ecoTILLING approach. Tilling provide excellent opportunity for tailoring crop plants that display desirable trait but lack powerful genetic tool. One example is given here, which was practiced or which was experimentized in rice. In rice, submergence tolerance gene sub1 was cloned and named as sub1 locus, that is sub1C, which was consistently expressed at a high level in submergence tolerance varieties of rice than in submergence tolerance rice, and transgenic ectopic expression of sub1. A in rice cultivar is lacking, it resulting in reduced expression under submergence, that is sub1C. In addition to providing an outstanding demonstration of the role of single new SNP in a key transcription factor can have on rice submergence tolerance, this approach also raises the interesting question of role played by sub1C in submergence tolerance or intolerance. This question can be addressed by tilling to identify inactive allele of sub1C in a japonica rice background that lacks sub1A. That means sub1C made a tremendous contribution for the development of submergence tolerance rice cultivar, but this is lacking in the japonica background where it can be found in indica background. So to identify what is the exact reason that it is not found in the japonica cultivar can be addressed by the tilling approach, because by just inducing single point mutation or just by deleting or knocking out or silencing that particular gene sub1C, we may go for identifying the function or why it is not activated or not functioning in the japonica kind. Tilling can be used for sequencing several complete plant genomes, or EST sequencing from many diverse plant species provides new opportunity to apply knowledge gained from functional genomics in rice and Arabidopsis. Identification of candidate orthologous genes is made easier by this particular approach. Smaller scale microcollinearity between genomes, if available, is most useful for transferring the positional information to the cloning of candidates orthologous genes. That means orthology between the genomes, maybe it may be mice genome or human genome, we can find the collinearity or microcollinearity. This can be a potential tool to demonstrate the utility of gene synteny and sequence homology in candidate gene identification. Using gene-specific primers, tilling can be applied to identify genetic diversity in candidate genes. That means maybe in wheat where ABD homologous genes for further study. EST sequence of wheat genes can be compared to rice gene structure to identify likely position of intronic regions for development of gene specific primers. That means tilling is utilized for identifying or generating the gene specific primers. Mainly, to talk about the applications of tilling, we may classify them into three strategies. One is under functional genomics, then genetic engineering, and third is evolutionary biology. We'll talk one by one. First is functional genomics, where identification of numerous mutations in target regions of genome. Construction of tilling library is useful for scientists to search for mutations in gene of interest. Tilling offers a way to investigate target gene of interest in any crop of interest without first having knowledge of gene product. Means prior sequence information is not required for tilling approach, where we go for sequence at the end. Second approach is genetic engineering, where we may go for foreign DNA of any type into plant genome. tDNA or transposon insertion are used to obtain specific gene knockouts, but practically limited to some crops only. Tilling is in front of transgene as consists of identification of numerous mutations within a targeted region of whole genome. Third is evolutionary biology, where alternative to wild relatives, tilling is used to introduce useful genetic variation in the allied germplasm, also applicable in a population which has several pre-existing polymorphism for developing SNPs. Coming to the advantages of tilling technology, its applicability to virtually any organisms means there is no boundary between the genomes or families or anything. It facilitates high throughput and independence of genome size, reproductive system, or generation time. Since chemical mutagenesis used here, all genes can be targeted by screening few individuals. High degree of mutational saturation can be achieved without excessive collateral DNA damage. ecoTILLING, इसके बारे में हम आगे बात करते हैं. दिस इज यूजफुल फॉर एसोसिएशन मैपिंग स्टडी एंड लिंकेज डिसइक्विलिब्रियम एनालिसिस. ecoTILLING can be used not only to determine the extent of variation but also to assay the level of heterozygosity within a gene. Coming to the merits and demerits. Merits of tilling: it's a cheap and rapid reverse genetic approach. Firstly, used to detect the chemical mutagenesis at single nucleotide level, independent of genome size, reproductive system, or generation time, valuable for essential gene. Demerits: skilled labor required, depend upon primary design, lower rate of induction of mutation. There are many scientific challenges before tilling, because as the tilling approach is a induced mutagenesis process, here scientists are facing a number of challenges like high quality mutant population. About 2 to 3 years is required. First step to generate a population is to use different concentration of the chemical mutagen to assess lethality to find an optimal concentration for inducing the experiments. It is challenging because the lethality of species and varieties respond differently to physical or chemical mutagenesis. Creating mutant population in vegetatively propagated plants is quite difficult. Species that are highly heterozygous may confuse mutation detection for researchers due to natural polymorphism. Then, production and maintenance of clones of vegetative propagated plants for future analysis are somewhat problematic. After the creation of mutant population, it is essential that all DNA extracts be equipped in concentration so that they are all correspondingly characterized in the pools being investigated. Otherwise, unique induced mutations may not be recognized as the amount of mutant DNA decrease in contrast to others in pool of DNA. Another task is particularly in plants in the selection of target genes that sometimes exist as a single copy throughout the genome. This leads to a problem when experiments on polyploid plants who have complex genomes like wheat and peanut. To overcome this contest, primer need to be designed that is precise to single gene of interest, which may entail some extra effort. That means utilizing tilling approach in polyploid species is quite difficult. Sequencing multiple alignments of the homologous target genes to find restriction site differences between the target genes. Proceedings to tilling the DNA can be digested, which may cleave the annoying target, leaving the desired gene unbroken for analysis. That means sometimes the heteroduplexes are unbroken so that the exact mutant cannot be identified. Potential difficulty in tilling may be created in the number of SNPs per fragment. Identification, scoring, tracking of the cleaved fragment hence becomes more challenging since SNP discovered in the heteroduplex need a high focused as compared to multiple mismatch sites. Care should be taken during scoring fragments as large number of SNPs are existing in a gene portion. That means being or having more number of SNPs in that particular genome may be confusing. Designing ecoTILLING or tilling experiment selection of the nucleus is very important to digest the mismatches in the heteroduplex. Cell 1 identify and cleaves the mismatch fragments in heteroduplex also contains 5' to 3' exonucleolytic activity can as well digest the full length PCR product starting with 5' fluorescent label. Therefore, care should be taken not to overdigest the DNA sample to avoid the loss of fluorescent signal of the PCR product. The most important challenge is allocating a particular phenotype to a genotype and supposing the putative function of a gene. Chemical mutagenesis sometimes create background mutations which can make phenotype analysis more difficult. This may take several generations of outcrossing and backcrossing. Obviously, to assign a function to a gene will be more challenging if there are any epistatic or pleiotropic effect created from the background mutations. That means presence of pleiotropy or epistasis are like villain in tilling approach. Coming to ecoTILLING is just a modified part of tilling. It is just like as we talk about linkage and association mapping, it is similar to that, where we talk about a particular population, means breeding population we use in linkage, but in case of association mapping we harness the natural variation. That means we take the natural population already existing. Likewise, ecoTILLING, it is a modified approach of tilling. So just we'll talk about some historical perspective. The first publication of ecoTILLING method, which is a modified form of tilling to for mining the natural polymorphism, was made in 2004 working with Arabidopsis thaliana. Here the exception to tilling in ecoTILLING is objective is to identify natural genetic variation as opposed to induced mutations. As you are knowing that in case of tilling we use EMS for inducing the mutation, but here we take the natural genetic variation to identify a mutation. Many species are not amenable to chemical mutagenesis, so ecoTILLING can aid in the discovery of natural variants and their putative gene functions. This approach allows one to rapidly screen through many samples with a gene of interest to identify naturally occurring SNPs and small indels. You may get a question in exam about ecoTILLING, why it is utilized. If you will get it in the bits, you need to answer with this thing. It may be either harnessing the natural genetic variation or to find out the SNPs or small indels. We'll discuss about ecoTILLING. This is an approach which is mainly utilized for allele mining. Allele mining means finding the rare alleles from the natural population or natural genetic variation is harnessed here. ecoTILLING is expected to provide a series of alleles for those genes that are involved in important processes of plant, even though known variants for these genes have not been observed through genetic studies. ecoTILLING is used to discover the catalog common polymorphisms without sample pooling, as was done for Arabidopsis and black cottonwood, that is Populus trichocarpa. To detect homozygous polymorphisms, an equal amount of DNA from reference sample is added to each assay. Here, when rare nucleotide polymorphisms are sought, samples can be pooled, as we have recently illustrated in using ecoTILLING to discover rare SNPs in human genome. This approach was done by Till in 2006. This is how tilling differs from ecoTILLING. The flow diagram is given here. The initial steps are same, like raw data is taken, filtration, compilation of the reference genes, mapping, variation calling. In case of tilling, what we do, we remove all the variations except CT and G2A. Base composition ratio study and variation selection is done, then represent variation in metrics. Whereas in ecoTILLING, the EMS, genetic EMS variations means induced variations are identified instead of removing the variations. Then removing the non-specific variations and the variation matrix is represented. The utility of ecoTILLING for screening many individuals at one locus, not good for few individuals at many loci, discovering rare haplotypes, and good in heterozygous population. Coming to the methodology, we shall just discuss, as I have already told you, how the tilling differs from ecoTILLING, but for better understanding, the pictorial representation is given here. I'll explain you clearly, just pay your attention here. In case of tilling approach, here you may find here, in tilling M0 generation, where EMS or induced mutagenesis is given, M1 is generated, self-pollination is followed, then M2, again self-fertilization, store the seeds. After growing that seed, particular seed, either in M3 or M4 generation, where the homozygous condition is obtained, then you go for pulling the DNA from those self-fertilized plants of M3 or M4 generation. Then you go for just designing the forward and reverse primers. Go for PCR. Identify the homodimer or homoduplex or heteroduplex. Then use cell 1 for digesting the heteroduplex. Run it on a gel slab or gel electrophoresis. Then sequence it and just see and identify the mutagenized population, or at which site the particular mutation means. Just identify whether mutations are present in that particular population or not. But what happens in case of ecoTILLING? Here you may find a natural population is taken. Then that population is carried forward. That means the plants are taken from there. We go for phenotypic observations. Wherever the mutant kind of plants are observed or some kind of variation is present, we will just take those plants, we'll go for pooling that particular DNA, then go for sequencing. After following the sequencing, gene specific primers are taken with two different composition here, or two different sequences here. Then you follow the same procedure, like identifying or going for PCR, sequencing the mutant parent, then homodimer and heterodimers are taken here, and those are ran in gel slab. Then go for identifying whether the mutant is present or not. That means the only difference between tilling and ecotilling is for identifying the SNPs or the natural variation existing in the population, whereas tilling is an induced approach. By inducing the mutant, we are finding whether particular mutant or changes are present in that particular population or not. I hope this is clear to you. So let us move to next slide. The third approach is detilling. So far we discussed about tilling, ecotilling. The third is detilling. This is a knockout approach. Knockout means kicking out. बोलते हैं ना kick out करो. That means just throw it away. Fast nutrient mutagenesis often results in kilobase scale DNA deletions. As a new knockout technique to obtain deletion mutant for target genes, strategy to screen for rare deletion mutants in large fast nutrient mutagenized populations was first developed by Lee in 2001. याद रखिए deTiLLING अप्रोच को दिया था ली एंड कोवर्कर्स ने 2001 एंड डेमोन्स्ट्रेटेड इन अरेबिडोप्सिस एंड राइस. आपका नॉलेज रिफ्रेश करूंगी कि ecoTILLING was also practiced in Arabidopsis. Tilling भी ऐसा ही है. Then it combines fast neutron mutagenesis and high throughput PCR screening named as delete a gene. This strategy has been further developed and named as deletion tilling or deTiLLING. Methods क्या क्या होते हैं इसमें? The steps involved are fast neutron mutagenesis, which generates DNA deletion of different size, DNA pooling strategy to reduce the number of PCRs needed, then technologies that allow a mutant alleles processing and internal deletion to be amplified in pools with excessive genomic target sequence or wild type alleles. देखिए deTiLLING कैन बी कंपेयर्ड विथ टिलिंग. ठीक है, ecoTILLING के साथ ये कंपेयर नहीं हो सकता बिकॉज इन ecoTILLINग वी हार्नेस द नेचुरल वेरिएशन एंड वी गो फॉर आइडेंटिफाइंग द SNPs. तो deTiLLINग एंड टिलिंग में क्या होता है? हियर इंड्यूस्ड म्यूटाजेनेसिस इज यूज्ड. टिलिंग में EMS यूज करते हैं एंड deTiLLINग में फास्ट न्यूट्रॉन यूज करते हैं. टिलिंग में क्या होता है? यहाँ पे कोई डिलीशन नहीं होता. हियर जस्ट चेंजेस आर इंड्यूस्ड और इंट्रोड्यूस्ड. बट इन केस ऑफ deTiLLINग नॉकआउट, जीन नॉकआउट और किलोबेस स्केल पे DNA डिलीशन किया जाता है, जिससे कोई भी डिलीटीरियस फंक्शनल अलील्स प्रेजेंट है या डिलीटीरियस जीन्स प्रेजेंट है, इसको हटाया जाता है. दैट इज ऑल अबाउट deTiLLINग. Friends, so far we discussed about tilling, ecoTILLING and deTiLLING. These are potential reverse genetic approaches in order to induce the mutations in a population. So I hope this information furnished by me is helpful for you. If you like this video, please do not forget to like, share, subscribe and keep on commenting. And your small comment or your small encouragement with subscription may motivate me for making further videos. Thank you once again.